So let's try to look at something different apart from what we've actually just covered in mathematics. And I happened to get a question from one of the students in our Kenya medical training institutions, and the student was so much interested in getting the difference between rods and cones. And I would actually be able to explain it in a very detailed manner for you to get a better understanding of the difference between those two. So, rods and cones. First, you should be able to understand that these are very important photoreceptors. So these two are photoreceptors. So photo means light, a receptor means something that perceives, something that receives, something that will accept, something that takes in, something that is to a greater extent sensitive to, for example, light. So rods and cones are photoreceptors. They perceive light. When light falls on them, that brings about a drastic metabolic or biological change that can bring about perception. So we need to ask ourselves, where can these rods and cones as photoreceptors be found? Rods and cones as photoreceptors are found on a very important surface of the eye called the retina. So I'll just be able to give a brief drawing of an eye of a human being, to be very specific, and where we can be able to locate these rods and cones. So assuming this is an eye of a human being, I'm so poor in drawing, so don't worry about my drawing. So just try to look at what I'm going to come up with so that you can be able to understand. Yeah, more specifically, I'll be so much interested in looking at where these rods and cones are found on the retina. And yeah, we can have this. Okay, so we will also be able to have this. Okay. Don't mind so much about the diagram, so so we can be able to have this part. Okay, yeah. So we'll have this. Yeah. Correct. So I would actually be able to call this as the conjunctiva, and this is referred to as the cornea. And I would actually be able to have this as the aqueous humor. I can have this. This part is what we refer to as the pupil, the opening that allows light into the eye, and the part that is just adjacent to it is referred to as the iris. It is very pigmented. For your information, I'll actually be able to give more information about it. And we can be able to refer to this as the ciliary body. Okay, and this can be referred to as the suspensory ligaments. The suspensory ligaments that will actually hold the lens in position and be able to bring about drastic changes in terms of the size of the lens, of the curvature of the lens, so that the lens can actually thin or actually be able to become thicker to actually determine how much light we can have getting refracted on our retina. So this is our lens, and this outermost part is the one referred to as the sclera. The one just close to it as you move into the eye is what we call so this can be our choroid, and this can be now our retina. This is what we refer to as the blind spot. There's a part here on the retina that we can refer to as fovea centralis. Fovea centralis. And some people call it the yellow spot. fovea centralis, or the yellow spot. Fovea centralis, or the yellow spot. Just on the retina. This is what we call our optic nerve that leaves the eye to the brain, more specifically the cerebrum, as the largest part of the forebrain. And of course, this space is what we refer to as the vitreous humor. So I'll actually be able to just give a brief description of the roles of each and every part of the eye before I can be able to get back to the question. So the conjunctiva is actually the part that appears to be on the outermost side of the eye, just in front of the eye. It actually tends to just be able to have the cornea lying below it, and this part prevents just dust particles or any other foreign particles finding their way into the eye. It appears to the inside of the eyelids, protecting the inner structures of the eye from any mechanical injury and damage and entry of any foreign particles like the dust particles. Just try to imagine you are moving, or you're just walking on a dusty road and a lorry passes by you, raising all the sand from the ground. You'll actually close your eyes, your eyelids will be able to actually flap over the surface of the eye, with the inner parts of the eyelids having the conjunctiva and preventing entry of those dust particles into the eye. The cornea that just lies below it is actually able to further also prevent entry of disease-causing microorganisms like bacteria. It can further also be able to prevent entry of any foreign particles like the dust particles into the eyes, and it actually nourishes the surface of the eye, nourishing in terms of actually ensuring that the eye remains moistened, and not nourishment in terms of providing nutrients. No, it ensures that the further the surface of the eye is from the conjunctiva, and the nearer it is to it as the cornea I'm actually talking about, the better it is in terms of having that moisturizing effect on the surface of the eye. The cornea contributes to the refraction of light. When light moves from an object, let's try to imagine I'm a human being and I'm looking at this girl. When light passes through the cornea contributes to the refraction of light so that all the light can fall on the retina for the purpose of perception. The cornea is also transparent to allow the passage of light through it, so that that light can be able to actually get refracted onto the retina for perception. Just after the cornea, as you move into the eye, you have what you call the aqueous humor. The aqueous humor has a nourishing effect in terms of providing nutrients to the cornea and the lens. Why? Because the lens of a human being, in terms of the eye, and the cornea do not have any supply of blood. And that means their nourishment and their ability to receive nutrients and oxygen is done by the aqueous humor. So the work of the aqueous humor is a water-like or a fluid-like substance that tends to supply nutrients dissolved into it to the cornea and the lens. Now, just after the cornea and the aqueous humor, we have what we call the pupil. The pupil is that opening into the eye. It allows light from an object to pass through it before reaching any other structures within the eye. For your information, any light passing through the pupil will either get refracted on the retina or refracted onto other parts of the retina. But for complete observation, there are structures within the eye that ensure that all the light is refracted onto the retina. More specifically, the most sensitive part of the eye, which is the fovea centralis, or the yellow spot. The iris is that pigmented part that surrounds the pupil. So you can be able to observe a tiny opening in the eye of your friend or relative. Around that tiny opening, there's actually a pigmented part. That is what we refer to as the iris. The iris first is pigmented, and that enables it to determine the color of the eye of an individual. So the color of an individual being black or blue is determined by the iris, which is pigmented, colored. The same iris is actually just a muscle-like structure. It has two very important types of muscles. The muscles are actually called circular and radial muscles. This can be a very good example. Let's talk about this being the pupil and this being the iris, having two different types of muscles. So what you call the circular muscles, they're of course circular, and the radial muscles, they tend to appear like the radius of a circle. So those muscles can rhythmically contract and relax antagonistically. Some contracting while others relaxing to actually alter the size of the pupil so that the amount of light entering the eye is regulated to prevent excessive entry of too much light into the eye that can damage the delicate structures within the eye. So you having your pupil becoming smaller or becoming widened to allow in either more light or less light is regulated by the radial and circular muscles forming the iris. So the iris one is pigmented to give the eye its color. Then number two, the iris has circular and radial muscles that are able to ensure that the pupil's diameter is altered, regulating the amount of light entering the eye. I have the lens. The lens of a human being are kept in position and suspended by the suspensory ligaments attached to the ciliary body. What you need to know is this. The lens completely refracts light to the retina. It is one of the most important structures that can be able to contribute to the refraction of light, moving from outside to the retina. Any unexpected changes to the lens can actually result into a blurred image or an unclear image getting formed on the retina. And the lens can actually end up developing a change in its size. So, for example, the curvature of the lens can reduce or increase. The size of the lens can reduce or increase. So there's what you call thinning, okay, for information, or bulging. And that all depends on whether an individual is looking at an object which is nearby or a far object. So that tells you that when a nearby object is observed or a far object is observed, there must be a change in the size of the lens in terms of its curvature so that a clear image is formed on the retina. So the lens can thin or actually get bulged so that either more light or less light is refracted, or there's complete refraction or there's less refraction of the light entering the eye for the purpose of accommodation of either a nearby object or a far object. So the lens contributes to the refraction of light to the retina for the purpose of perception. It can get altered in terms of its curvature to accommodate a nearby object or a far object during the observation. Now, the suspensory ligaments are the ones that actually end up changing the size of the lens. And these ligaments, one, will hold the lens in position. Two, they either become taut or they become relaxed. And this will all depend on what the ciliary muscles can do. When ciliary muscles act on the suspensory ligaments, or there's less action of the ciliary muscles on the suspensory ligaments, these ligaments can either become relaxed or they actually end up experiencing more tension, and that can make them to change the size of the lens. So ciliary muscles can act on the suspensory ligaments. Suspensory ligaments can either relax or encounter more tension, and that can make them to actually alter the size of the lens or the curvature of the lens. And that is to say, the ciliary muscles will actually act on the suspensory ligaments, making the ligaments to become tensed or relaxed to bring about a change in the size of the lens for the purposes of accommodation of a nearby object or a far object. The ciliary body first secretes the aqueous humor. The ciliary body first secretes the aqueous humor that nourishes the cornea and the lens, as per my previous discussion. The same ciliary body will be able to actually facilitate the formation of ciliary muscles, which are now specialized to facilitate the change in the size of the lens indirectly when they act on the suspensory ligaments. Just near to that is what you call the vitreous humor. Is a jelly-like substance that contributes to the refraction of light to the retina. So when light passes through these parts, this vitreous humor contributes to the refraction of light so that all the light can fall onto the retina. So the first part to actually refract the light is the cornea. The second part that also contributes to the refraction of light is the aqueous humor that still nourishes the cornea and the lens. The second part that further refracts that light is the lens. The other part that can contribute further to the refraction of that light is the vitreous humor. This is actually a watery or fluid-like substance. This is a jelly-like substance or mucus-like substance. We've actually ensured that all the light has actually fallen on the retina for the purpose of perception. As you move further, you'll realize that the outermost part just behind the eye is called the sclera. The sclera is made up of cells, and those cells are able to protect the delicate inner structures of the eye. So one of the most important functions of the sclera as the outermost part of the eye is to protect the delicate structures of the eye from any mechanical injury and damage. Just lying below it is the choroid. The choroid is also pigmented. The work of the choroid is to actually be in contact with the retina, such that when light reaches on the retina, it absorbs any stray light, so that no blurred images or not so many images are formed in the eye. So any light that seems to be diverted, any light that seems not to fall on the retina, any stray light reaching the eye can get absorbed by the pigmented choroid. Any stray light that reaches the eye and not falling on the retina can get absorbed by the choroid to avoid forming blurred or unexpected or many images within the eye, so that only one image is formed on the fovea centralis or yellow spot as the most sensitive part of the retina, with more cones to facilitate perception. Choroid and sclera can extend themselves to move and be able to form some structures just at the front part of the eye. They've actually said this. If you look at the choroid, it actually ends up forming the ciliary body. The ciliary body can form the ciliary muscles. The ciliary muscles can get attached to the suspensory ligaments. If you look at the sclera, it ends up forming the cornea. And of course forms part of what we call the sclera, just at the front part of the eye. That is something you're supposed to actually understand in a deeper way. As you move just to the inner parts of the eye as well, you'll realize that whatever gets attached on the choroid to the inner part is what we call the retina. And to get back to our discussion, you'll realize that the retina is the one that has what you call the rods and the cones, which are photoreceptic. They are very sensitive to light. When light falls on them, they actually facilitate the transmission of a nerve impulse generated. And because of what the question was actually all talking about, I'll mostly focus on how those rods and cones can bring about perception. And I've actually told you this. They are found on the retina. They form the retina. Those rods and cones, they form the retina. And those rods and cones are found at different parts of our retina. and their concentration differs as well in different animals. Remember, we have animals called nocturnal animals and other animals called diurnal animals. Nocturnal animals can have a better perception at night, such as cats and even dogs, and those animals have more rods than cones. If you come to human beings, we are in a better position to perceive things during the day when there is more light, and that means we have more cones. So we do tend to have less rods, but the number of rods can actually be able to increase in nocturnal animals. So we are actually diurnal animals. Diurnal animals are those animals with more cones and less rods. They are in a better position to perceive things better during the day and not be able to perceive things well at night. While nocturnal animals can be able to better perceive things or objects in the environment at night than they can do during the day. So let's get into our discussion. So you should understand this, that if I do really want to have a better understanding of this, I can have this as my retina. And for information, the retina has three very important regions that you really need to understand how they work. This innermost part, we can talk about the outermost part, is the one in contact with the choroid. I told you the outermost part is the sclera from our previous discussion, and this part just close to the sclera is what you call the choroid, which is pigmented to absorb any stray light reaching the eye. Then just from the sclera we have to move to the choroid. Then from the choroid we have to move to what you call the retina. So that part, which is now the outermost part for your information, that is in contact with the choroid, forms part of what you call the three main regions of the retina. The middle part of the retina has what you call nerve cells or sensory cells. That's the middle region of the retina. To mean, the retina has three regions. The outermost region in contact with the pigmented choroid. The middle region having sensory cells or nerve cells. Then the innermost region having now the rods and the cones. You might get this question one day. Someone can ask you to give the three main regions of the retina. The retina has three regions. The first region is the outermost region in contact with the choroid, the pigmented part of the eye, absorbing stray light. The middle region has sensory cells that get triggered when light falls on the rods and cones so that a nerve impulse is generated and transmitted. Then the innermost region of the retina has the photoreceptor cells called the rods and cones that are sensitive to light when light falls on them. So let's just try to look at the differences between these rods and cones and where they can be actually found in the animals that do have eyes. For your information, you should know that one, this can be a brief diagram. So let me talk about this. So we have this can be our first diagram or second. I don't mind about my diagrams as long as you understand the concept. So you can have this. Okay. Then you can have this as well. So you can have this as well. You can have this as well. So a keen look at this will actually tell you that This is just my simplest diagram to explain this concept. So a keen look at this can tell you that this can probably be our rods, and these are going to be our cones. I'll explain these things in detail for you to understand the differences between these two very important photoreceptor cells in our eyes, forming the innermost part of our retina. And without them, perception could not be possible for information. They are very sensitive to light. So let's actually begin with the cones. Cones, as I've actually told you, are mainly found in diurnal animals. In other words, they are actually able to get found in animals that can perceive better during the day. So animals that can perceive better during the day. Animals that can perceive better during the day. Okay, perceive better during the day. But rods are mostly found in, correct, can use this. So mostly found in nocturnal animals. So these are animals that can perceive better during a time when darkness has actually taken over. Perceive better at night. Perceive better at night. And let's proceed. I want to actually mention something else which is very important. If you look at these cones, this probably, as you can look at it, is the first cone, the second cone, and the third cone. And each cone is connected to its own bipolar neuron. Now, what am I trying to communicate here? In as much as you may want to actually mention about sensory neurons are unipolar. To mean they actually end up having their own cell body of the axon. But for the purpose of understanding this, we can better change this so that we have what you call a bipolar neuron. A bipolar neuron. Instead of having a sensory neuron in our discussion, you can have what you call a bipolar neuron. And I would actually understand that any discussion in biology that has to get mentioned as far as this part should actually focus more on the ability of a particular nerve cell getting mentioned, being able to get given the right features. For example, if you look at this cone, it has its own bipolar neuron. A neuron is just a nerve cell along which a nerve impulse can get transmitted from one part of the body to another. For example, from a particular receptor organ to the central nervous system, or from the central nervous system to an effector organ at a particular point in the body of a human being or an animal. So why do we call this a bipolar neuron? Because there are two dendrites projecting from this cell body. This is what we call the cell body. So sensory neurons may not be so much discussed here, although they still can allow nerve impulses to get transmitted from the receptor organ to the central nervous system. So each cone has its own bipolar neuron. Why do we call it a bipolar neuron? Because I've actually said from the cell body, we have what we call two dendrites. Bi means two. So from the cell body, we have two dendrites projecting from it. And when two dendrites project from the cell body, so this is our cell body, this is the first dendrite, this is the second dendrite. So two dendrites are projecting from the cell body. That gets called a bipolar neuron. So each cone is able to get connected to its own bipolar neuron. Each rod does not get connected to each own bipolar neuron. But here, each cone with its own bipolar neuron, each cone with its bipolar neuron, each cone with its bipolar neuron, before they can actually be able to get joined to the same optic nerve that can now carry that nerve impulse to the brain for interpretation. So here, you can say there is no retinal convergence. Here, there is retinal convergence. Why? The three rods are connected to the same bipolar neuron. So there's what we call convergence. Three rods are connected to the same bipolar neuron, which can now transfer the impulse to the optic nerve. But here, each cone has its own bipolar neuron, so there is no retinal convergence. So whenever each cone has its own bipolar neuron, then there's something called high visual acuity. So cones have no retinal convergence, so they have high visual acuity. That means have ability to distinguish objects. Have ability to distinguish objects clearly. I can look at this and say this is a woman from Nyeri because she has a light skin and she is of a small body size. This is a woman from Kakamega. She has a larger body size and she is dark skinned. So those detailed features that can get found in two objects which are close to each other can get brought out very clearly when cones are under work. Why? Because they have no retinal convergence and each cone has its own bipolar neuron. Look at that. So no retinal convergence in cones can bring about high visual acuity or the ability to distinguish objects clearly, bringing out the finer details in each object. Bringing out finer details in each object. Here is what you call low visual acuity. Low visual acuity. Low visual acuity. So this is what you call low visual what? Acuity. Low visual acuity. So to mean inability to distinguish objects. Ability to distinguish objects. Okay, something else. Cones have a photochemical substance called iodopsin. That is to tell you that each cone has a chemical in it. That chemical is the one that makes that cone to become sensitive to light. So that photochemical substance. So a photochemical substance is called iodopsin. But rods have a photochemical substance called rhodopsin. So in each cone, there's a photochemical substance sensitive to light, and that chemical is called iodopsin. In this other case, the rods have a photochemical substance called rhodopsin. Photochemical substance called rhodopsin. Okay, as you move forward, you'll realize that when light falls on a cone, the iodopsin splits. The iodopsin breaks down. The iodopsin disintegrates to give us iodine and opsin. It's a complex protein. When light falls on a cone, the iodopsin chemical in that cone, that photochemical substance, that chemical sensitive to light in that cone, will get split, will get broken down, will get disintegrated to form iodine and opsin. Opsin is a complex protein, and when that compound splits or breaks down, the sensory hairs in the middle region within that retina will be able to get stimulated, will get triggered, and thus a nerve impulse is generated. That nerve impulse that gets generated is transmitted along the optic nerve to the cerebrum part of the brain responsible for vision. Let's get to rhodopsin. Rhodopsin will actually be able to get found in every rod. And when light falls on a rod, the rhodopsin as a photochemical substance in that rod will split into retinine. Retinine is a derivative of vitamin A. Derivative of vitamin A. A derivative of vitamin A. So if I go to vitamin A, I need to find a substance called retinine. That retinine is very important for perception. That's why sometimes you'll get a question actually asking you, name the type of vitamin that can cause night blindness, inability of an individual to perceive objects well at night. And remember, I've told you, rods are mostly found in nocturnal animals, those that are better able to perceive things well at night. So night blindness is the inability of an organism to perceive things at night. And that means one of the causes can be lack of enough vitamin A. When there's lack of enough vitamin A, then a derivative called retinine should be lacking in that rod. When the rods lack retinine, then that means they lack rhodopsin. And when rhodopsin is absent in that rod, the rods cannot be able to perceive light. And that means there will be the inability of that individual to perceive that object or those objects at night. So rhodopsin is a chemical found in every rod. Rhodopsin consists of retinine. And where can we get retinine? In vitamin A. So when I take in foods rich in vitamin A, I'll be taking in retinine. Retinine will contribute to the formation of rhodopsin. Rhodopsin is a very important chemical responsible for perception of objects at night. So take note of that. Apart from retinine, a derivative vitamin A, this same rhodopsin can also be broken down into opsin. The same opsin here, which is a complex protein that can also contribute to the formation of these very important photochemical substances in both rods and cones. Now, to proceed forward, you should understand this, that these cones are majorly three. And these three cones are able to actually become very sensitive to color perception. Very sensitive to color perception. We have three cones, and this is very, very common in higher vertebrates. Higher vertebrates like human beings. So if you go to higher vertebrates like human beings, there are three main types of cones responsible or sensitive to color perception. So cones are three mainly in higher vertebrates like human beings. And those three cones are responsible for color perception, sensitive to color perception. So they consist of the red, the blue, and the green. The red, the blue, and the green. They are responsible for color perception. Responsible for color perception. So there are three: red, green, and blue. And they're very, very important during what you call color perception. You'll just try to imagine, for example, if I decide to actually look at these three cones and they help me to actually perceive color. Someone told me that when the red cone or the cone with a red color perception and that other cone with a green color perception are equally stimulated. So light falls on the red cone from an object and light again falls on the green cone from an object. When they all get equally stimulated, then a yellow color is perceived. So red and green cones all equally getting stimulated can bring about yellow color perception. And someone somewhere also told me that when both the red, blue, and green cones are all stimulated, a sensation of white color is perceived. I repeat, when both the red, blue, and green cones are equally stimulated, and a nerve impulse is transmitted along the optic nerve to the brain, then a white color sensation is actually encountered. So red, green stimulation can bring about yellow color perception, and red, blue, green stimulation equally at the same time can bring about a white color sensation. So cones are sensitive to light, and they are very sensitive to light, and they end up bringing about color perception. So these rods, also referred to as visual purple, are less sensitive to color perception, are less sensitive to color perception, are less sensitive to color perception. So they are less used in terms. So opsin will always be a complex protein, just like in the same way this protein is complex in as far as the cones are concerned. And you've talked about the retinine, a derivative of vitamin A, and you've actually mentioned that rods have a visual purple color perception, but they are very less sensitive to color perception. So most rods do not actually help in color perception, but the ones that actually get helped in color perception are the cones. You've said there are actually three. The red and green can equally be able to get stimulated to bring about the yellow color perception. And if both red and blue together the green are all stimulated, then they can bring about white color sensation. So in higher vertebrates like human beings, you'll find more cones which are very sensitive to color perception. That is something very, very important. So to just add on to this, there's something called trichromatic theory. Trichromatic theory. Just sketch trichromatic theory. So rods and cones. So in cones, there's something called trichromatic theory. So this states that there are three cones for color perception, and I've actually told you they are the red, blue, and green. And this theory states that if the three cones for color perception are all equally stimulated, if the three cones for color perception are equally stimulated simultaneously at different degrees, then color perception is brought about. I've said rods are very less sensitive to color perception, but cones are very sensitive to color perception. And there are three main types of cones responsible for color perception: the red, blue, and green. I've even mentioned and said that when red and green are equally stimulated, they bring about yellow color perception. But if all the three, the red, blue, and green, are all equally stimulated at different degrees simultaneously, they'll bring about white color sensation. So trichromatic, tri means three, so the red, green, and blue. Chromatic, of course, chromatology is the study of colors, so chromatic from the word color. So if both the three, red, blue, and green cones for color perception are stimulated simultaneously at different degrees or at different extents, then color perception is brought about. And something else just to note about rods and cones is that rods are sensitive to low light intensity. That is why they are well used at night when there's low light intensity. But cones are sensitive to high light intensity. That's why they are well used during the day. But the good thing is this: in diurnal animals like human beings or higher vertebrates, there are both rods and cones to bring about an effective perception, both during the day and at night. But the concentration differs depending on the type of organism you're actually referring to. But rods are mostly found in nocturnal animals like cats and dogs, which perceive better at night and less effective in perception during the day. Where do we find rods in the eye? Rods are found at the periphery of the eye, just on the sides. That's why if someone finds himself or herself going into a house, maybe a room that is not well lit, or you're just checking for something just under the bed when it is dark, you'll tend to actually position your head in such a way that you'll really want to show that you are using something just at the periphery of your eyes, not directly just looking at something under the bed. You'll tend to actually open your eyes wide and lower your head and look like you're really struggling to look at something using a particular structures just at the periphery of your eyes. Just to the periphery of your eyes, you'll find the rods in large numbers. So on the periphery of your eyes, you'll find the rods. But now, cones are found. This must be mainly, cones are found at the yellow spot or fovea centralis. This is where the image is mainly formed with the retina. So the yellow spot or fovea centralis has a larger concentration of cones. This is where the image is formed with the retina of an animal that has the ability to perceive. Correct. So regardless of whichever situation you may end up having, both rods and cones are found in both animals, and they actually help the organisms to perceive things both at night and during the day. But there are those that can better perceive during the day, like the diurnal animals, and those that can better perceive at night, like the nocturnal animals, depending on the difference in the concentration of both the cones and the rods. There is a part of the retina called the blind spot. The blind spot is where the optic nerve leaves the eyeball. At that point, we do not have rods and cones. And at that point, as per my previous diagram, there are no images that get normally formed. So, any light falling at the blind spot of your retina will not end up forming any image. So the point where the optic nerve leaves the eyeball to the brain or the cerebrum, we do not have rods and cones, so any light falling at the blind spot will not be able to form any image. It's called blind spot because no image is formed. It enhances blindness since no image can get formed at that point. Thank you so much for your time and have a blessed evening. Let's meet in our next lesson.